Hello, my talk has three parts. I will begin by explaining the basic methods used for heritability analysis, in particular how to estimate SNP heritability, heritability enrichments, and genetic correlations. I will then describe something called the heritability model and explain why this is very important when performing heritability analysis. I will then finish with some recent extensions of heritability analysis that are being used to better understand and to predict complex traits. You are probably familiar with the idea of heritability. The heritability of a trait is the proportion of phenotypic variation due to all genetic factors. So for example, the variation we observe in human height is about 20 centimeters squared. Of this, we've estimated that about 16 centimeters squared is due to genetic factors, and therefore the heritability of height is about 80%. For SNP heritability, on the other hand, is the proportion of phenotypic variation explained by SNPs, single nucleotide polymorphisms. These are a basic type of genetic variant. For SNP heritability is important for a number of reasons, particularly for GWAS, genome-wide association studies. The aim of a GWAS is to find SNPs that are important for a phenotype. If a trait has high heritability, it means the SNPs are very important for a phenotype, and therefore a GWAS is likely to be more successful. This slide provides an overview of how we estimate total heritability. It requires related individuals. The basic idea is that if a trait is heritable, the more closely related individuals, for example brothers, sisters, parents, children, will tend to have more similar phenotypes than less closely related individuals and unrelated individuals. We've been able to estimate heritability for a long time. This figure shows some data collected by Francis Galton over 100 years ago. What he did was compare the height of children with the average height of the parents, and he observed that taller parents tended to have taller children, and therefore he concluded that height was a heritable trait. I will now explain two methods for estimating SNP heritability. These next four slides are quite complicated. I'll try my best to explain them simply, and then at the end I'll give a summary of the most important points. The first method is GCTA. This requires individual level data for unrelated individuals, so no pair of individuals can be more closely related than, for example, second cousins. First, some mathematical notation. Let X be a matrix of genotypes, so Xj contains the values for SNPJ. Y is a vector of phenotypes, and we assume that both genotypes and phenotypes have been standardized. This doesn't change any of the assumptions, but it makes the maths a bit easier to read. So GCTA begins by assuming this long linear model that Y is a linear combination of SNP values. So beta 1 is the effect of SNP 1, beta 2 is the effect of SNP 2, up to beta M, the effect of SNP M. So M could be maybe 500,000. There's also a noise term E. It then assumes that effect sizes are normally distributed. So beta J is normally distributed with variance sigma squared G, the genetic variance, divided by M, the number of SNPs. It also assumes that the noise is normally distributed with variance sigma squared E, the noise variance. If you make these three assumptions, then we get the Y is also normally distributed, and its variance has this form: K, a kinship matrix, times sigma squared G, the genetic variance, plus I, an identity matrix, times sigma squared E, the noise variance. GCTA solves this model using something called restricted maximum likelihood, from which we get the estimate of SNP heritability. It has this complicated form, but because we've standardized variables, we get that the estimate of SNP heritability is the estimate of the genetic variant. I will now explain the heritability model. So a bit more notation. Let H squared J represent the heritability contributed by SNPJ. So the total SNP heritability is the sum of all the H squared J's. The heritability model describes our prior expectations about how heritability is distributed across the genome, so it specifies the expected heritability contributed by each SNP. So what heritability model does GCTA assume? Well, the heritability contributed by a SNP takes this form. It's this effect size squared times its variance divided by the phenotypic variance, and because we standardize things, it's equal to the effect size squared. So hopefully this makes sense. If a SNP has a bigger effect size, then it contributes more heritability. Now, because we've assumed that effect sizes are normally distributed, we know their expectation, and so the expected heritability is the genetic variance divided by the number of SNPs. Or alternatively, it's the SNP heritability divided by the number of SNPs. So the point is that GCTA assumes that each SNP is expected to contribute an equal share of SNP heritability. The second method is LD score regression. Now this does not require individual level data, but instead only uses summary statistics and a reference panel. So let's suppose we perform single SNP analysis, and TJ is a chi-squared test statistic from regressing the phenotype on SNP-J. The expected value of TJ takes this form. It's 1 plus n for sample size times big H squared J, where big H squared J is the heritability tagged by SNP-J. So hopefully this makes sense. The test statistic of a SNP depends on how much heritability a SNP tags. It also depends on the sample size. Here's an expression for the heritability tagged by SNP-J. It's the heritability that the SNP contributes itself, plus how much of the heritability it tags from neighbouring SNPs. So here, R squared JK is the correlation squared between SNP-J and SNP-K. LD score regression assumes that each SNP is expected to contribute equally towards SNP heritability, so the same assumption made by GCTA. And if we make that assumption, then we can calculate the expected heritability tagged by each SNP. It has this form. It's for SNP heritability divided by the number of SNPs times LJ. Now LJ is a measure of tagging, how well SNP-J is tagged by its neighbours. It's also referred to as the LD score of SNP-J. And then further, we can insert this expectation in the equation on the previous slide and get the expected test statistic for each SNP, and that equals 1 plus the sample size times the SNP heritability divided by m times the LD score. So hopefully you can appreciate that now we've written the expected test statistic of each SNP in terms of just one unknown for SNP heritability. So if we regress test statistics on the LD scores, we can obtain an estimate of SNP heritability. So to summarise, there's two main approaches for estimating SNP heritability, GCTA and LD score regression. The main difference of these two methods is that GCTA requires individual level data, whereas LD score regression requires only summary statistics. But also, the main similarity is that both methods assume the same heritability model. In particular, they both assume that each SNP is expected to contribute equally towards SNP heritability. And I will be referring to this heritability model as the GCTA model. Both GCTA and LD score regression have been extremely popular. Each has been applied to thousands of different phenotypes. Here are some results using 14 traits from UK Biobank. For these analysis, I use the LD score method, so I use summary statistics and assume the GCTA model. We see that for all traits, there's substantial SNP heritability, at least 10%. For height, it's as high as 50%. And this reflects what we've seen across statistical genetics as a whole. These analyses have shown that SNPs are important for a wide range of complex traits and diseases, and therefore justify the GWAS approach. I will now describe some extensions of the method for estimating SNP heritability. There have been a number of extensions which have produced new tools for better understanding complex traits. I will describe extensions of the LD score method, but in most cases there's a directly equivalent extension of the GCTA method. All these extensions are obtained by generalising the heritability model. So one extension allows us to divide SNP heritability across the genome. So the top is the basic heritability model, which has one unknown. We will now slightly generalise this to have two unknowns. We will divide the total SNP heritability into H-squared genic and H-squared intergenic. That's the contribution from genic SNPs and the contribution from intergenic SNPs. And let's suppose there's M1 and M2 of each of these. So we now assume that the expected heritability contributed by a SNP is not constant across the genome, but instead, if it's within a gene, it's H-squared genic over M1, and if the SNP's not in a gene, it's H-squared intergenic over M2. It's a bit more complicated, but we can use the same process to obtain an expression for the expected test statistic of each SNP, and instead of having one unknown and one set of LD scores, it has two unknowns and two sets of LD scores. And then we can regress the test statistics we observe on these two sets of LD scores to obtain estimates of H-squared genic and H-squared intergenic. So here's an example. Instead of dividing SNPs based on genic and intergenic, they actually divided them based on chromosomes. So they used a 22-part heritability model, which allowed them to estimate the proportion of SNP heritability contributed by each chromosome. They analysed a variety of traits, and what they found was, in general, the heritability contributed by a chromosome was proportional to the length of the chromosome, and this demonstrated therefore that all these traits were polygenic. They had thousands of causal variants spread across the whole genome. Here's the second example, which used an extension of the LD score regression method. Now they divided SNPs based on functional annotations. In total, there were 52 different categories. They analysed a wide range of traits and estimated the heritability explained by each category. And from that, they estimated the enrichment, so how much SNP heritability a category explained, divided by its size. Here is a figure of their main results, and the biggest enrichments they observed were for conserved SNPs. These represent about 2.6% of the genome, but they estimated that they explained about 30% of SNP heritability, so about 13-fold enrichment. A second extension allows us to estimate genetic correlations. The genetic correlation is the measure of the genetic similarity between a pair of traits. So now suppose we have summary statistics from two traits, but instead of chi-squared test statistics, we'll be using Gaussian test statistics. Now, instead of estimating the SNP heritability, we're interested in estimating the SNP co-heritability, but the method is very similar to before. We first assume the co-heritability is evenly distributed across the genome, and that allows us to get an expression for the expected value of the product of these Gaussian test statistics for each trait. It has this form. It's a function of the total SNP co-heritability and the LD score. So similar to before, we can now estimate the SNP co-heritability by regressing the product of these test statistics on the LD scores, and that in turn allows us to estimate the genetic correlation. So here's an example. This paper used an extension of LD score regression to estimate genetic correlations across 24 different human traits. And what they found was that many pairs of traits had substantial genetic correlation, showing that many complex traits have a very similar genetic architecture. A final extension allows us to estimate confounding bias. The amount the GWAS test statistics have been inflated by factors such as population structure or familial relatedness. Now, we actually use the same heritability model as before, but instead we slightly change the expression for the expected test statistic for each SNP. Specifically, we add in this coefficient A, which represents the average amount that each test statistic is inflated due to confounding. But otherwise, the method is the same. We regress the observed test statistics on their expected values, and this then allows us to estimate not just the SNP heritability, but also A, the average amount of confounding in the GWAS. So here are some results from the original LD score regression paper. They compared these new estimates of confounding bias to those corresponding to genomic control. So many of you will be familiar with the idea of genomic control. It tries to correct for confounding by scaling all test statistics so that the new medium test statistic matches that expected if there was no causal variation. So this paper analysed a wide range of traits. Here are a few examples. And so for schizophrenia, genomic control estimates that each test statistic is inflated on average by 48%, whereas LD score regression estimated that the inflation was only 7%. And therefore they showed that genomic control tends to overestimate the confounding and therefore overcorrect. So far, I've only explained the GCTA model, in which each SNP is expected to contribute equally to SNP heritability. And although this is the most common, there are actually a wide number of other models which have been proposed. So for example, I proposed the LDAC model, and here the expected heritability of each SNP depends on two parameters, WJ, which is a measure of linkage equilibrium, and FJ, which is the MAF of the SNP. And so specifically, the LDAC model assumes that SNPs which are in regions of lower LD, or which have higher minor allele frequency, tend to contribute more heritability than SNPs which are in regions of higher LD, or which have lower minor allele frequency. So GCTA authors have also proposed a new model, the GCTA-LDMS model. The authors of LD score regression have proposed a new model, the baseline LD model. And I've also proposed a new model, this BLD-LDAC model. Now all three of these are quite complicated. Here I've written out the expression for the BLD-LDAC model. It includes 66 parameters and 66 SNP annotations. And in this model, the expected contribution of each SNP depends on LD, it depends on minor allele frequency, and also on functional classifications. It's important to realise that the choice of heritability model can be very important when performing heritability analysis. So in this example, I estimate SNP heritability for the same 14 traits as before, but now I compare estimates obtained assuming the GCTA model to estimates obtained assuming the LDAC model. And what we see is the estimates assuming the LDAC model are quite substantially higher, on average 40% higher than those from GCTA. The heritability model is also important when estimating heritability enrichments. So here we performed an analysis similar to that of Finucane et al., who used 52 different functional annotations. We compare in red are the estimates of enrichments obtained assuming the GCTA model, while blue are the estimates obtained by assuming the LDAC model. And what we see is the estimates assuming the LDAC model are generally much more modest. For example, under the GCTA model, conserved are estimated to be 9-fold enriched, whereas under the LDAC model, they're estimated to be only 2-fold enriched. One area where the heritability model doesn't seem that important is when estimating genetic correlations. So here we analysed 24 different traits and we estimated the genetic correlation between all pairs. And what we find is that the estimates of genetic correlation assuming the GCTA model, those on the x-axis, are highly similar to those estimates obtained assuming the LDAC model on the y-axis. However, this seems to be the exception. In general, the choice of heritability model is important when performing heritability analysis. So the next question is, how do you decide which is the most appropriate heritability model to use? So some groups have compared models based on simulation studies, but we're against this approach because the model that performs best in simulations will depend on the assumptions made when simulating the data. Instead, we strongly recommend that models are compared based on how well they fit real data. And so in 2017, we showed it was valid to compare models based on a model fit for log likelihood calculated from GREML. We analysed in total 42 different traits, and we consistently found that the log likelihood from the LDAC model was higher than that from the GCTA model, showing that the LDAC model should be preferred. While this is valid, it has limitations because GREML requires individual level data, and it's only feasible for simple heritability models. Therefore, in our most recent work, we proposed something called log-LSS, which is an approximate log likelihood that can be computed using only summary statistics and for all heritability models, no matter how complex. We use this to compare in total 12 different heritability models based on how well they fit data from 31 different GWAS, and this table shows a summary of the results. First, we compared models based on average log likelihood, but also based on the average AIC, which discounts the log likelihood based on how complex each of these models are. On both measures, the best performing model was this BLD-LDAC model. We also proposed a new model. We've called it the LDAC thin model. It's just a simplified version of the LDAC model, and that was the best performing of the simple heritability models. So now we recommend, where possible, all analyses use this BLD-LDAC model, but some analyses do require a simple heritability model, in which case we recommend using this LDAC thin model. So for completeness, I'd like to return to these estimates of confounding bias. So we found that not only are they sensitive to a choice of heritability model, but also to a choice of confounding model. So as a reminder, these estimates are based on the assumption that confounding affects all test statistics equally. In our paper from last year, we found examples where this assumption is strongly violated. But the problem is we can't think of a way how to test whether this assumption is valid, and therefore whether to know whether estimates of confounding bias are valid. And as a result, we now recommend that you don't estimate or allow for confounding bias in your analysis. And this means that therefore you should only use summary statistics when you're confident the GWAS has performed very careful quality control. I will finish by describing some recent extensions. The first is a method for estimating the selection-related parameter alpha. We assume a heritability model of this form, where the expected contribution of SNP-J is a function of Fj, its minor allele frequency, to the power of 1 plus alpha. So here alpha determines the expected relationship between effect sizes and minor allele frequency. If alpha is greater than 0, it means that more common SNPs tend to have bigger effect sizes, and vice versa. Now this model is fairly complicated, so we cannot solve directly for alpha, but instead we use a grid search. We test a range of alphas between alpha minus 1 and alpha 0.5 and see which version fits best. So here are some estimates of alpha for the 14 UK biobank traits. Now it's worth pointing out that the classical model of selection that proposed by Ronald Fisher corresponds to alpha minus 1, And this is also the assumption made by the GCTA model, that there's a very strong negative relationship between effect size and minor allele frequency. But what it's clear to see is the data do not support this value. While we do see evidence for negative alpha, significantly for about half the traits, the estimate is much more modest. Across the 14 traits, the average estimate of alpha is minus 0.25, and this is our recommended value for performing heritability analysis. So the results on the previous slide were estimates of genome-wide alpha, but we can also generalize this method to obtain estimates of how alpha varies based on functional annotations. So in total, we considered 24 different functional categories of SNPs, and for this, the red lines are estimates of alpha inside the category, the blue lines are estimates outside the category. And what we see is for a wide range of these annotations, there is evidence that alpha is much lower inside the category than outside. And so, for example, for coding regions and promoters, we see that alpha is closer to minus 0.5. And so that indicates that the impact of selection is much stronger on functional categories of SNPs. I will finish by showing how these heritability analyses can be used to improve our prediction of complex traits. So when we estimate SNP heritability, we also obtain estimates of the heritability contributed by each individual SNP. So for example, when we assume the GCTA model, if our estimate of SNP heritability is 0.5, then this means that our estimate of the heritability contributed by each individual SNP is 0.5 divided by m, the number of SNPs. Now individually, these estimates are very poor, they're very imprecise. But the fact is that these can be used to apply distributions for other analysis. And of course we can slightly improve these estimates by using more realistic heritability models. So these estimates can be used to improve the performance of existing prediction methods. So here we've considered 5 different prediction methods, hopefully you've heard of some of them: BLUP, LeSue, LDPred, Bayes-R and Bolt-LMM. Now the default version of each of these methods assumes that the expected contribution of each SNP is constant, i.e. the GCTA model. So we even said develop generalized versions of these where the user is allowed to specify the expected heritability of each SNP. So for example, BLUP, best linear unbiased prediction, assumes that effect sizes are normally distributed with a constant variance. So we've simply developed a version where the user can specify the variance of each effect size distribution. We've used each of these 5 tools to analyse each of the 14 traits from UK Biobank. First, we assume the GCTA model, i.e. the default versions. Then we assume the LDAC-thin model, which is a slightly better heritability model. And then we assume the BLD-LDAC model, which is our best heritability model. So here are the results. These bars report the correlation squared between predicted and observed phenotypes from each method, averaged across the 14 UK Biobank traits. In total, there's 5 blocks corresponding to the 5 methods, and then the colour indicates the assumed heritability model. So what we see is when we switch from the GCTA model to the LDAC-thin model, average prediction always improves for all of the 5 different methods. Then when we switch from the LDAC-thin to the BLD-LDAC model, it improves even more. And in fact, it's not just the average performance, actually the prediction improved for every method for every single phenotype. So these results are exciting, particularly when you consider the change in the heritability model has no impact on the computational demands of a method. So here we've taken the best performing method, Bolt-LMM, and we've varied the number of training individuals from 100,000 up to 200,000. So each line reports the average correlation squared for each heritability model. And what we see is changing from the GCTA to the BLD-LDAC model is equivalent to increasing the sample size by about 25%. And as I say, this is completely free improvement because changing the heritability model has no impact on computational demands. So to summarise, I've explained the basic methods for performing heritability analysis, whether using individual level data or summary statistics. Now, a key thing to realise is that all heritability analyses start by specifying a heritability model. And often, the choice of model is very important. It will have a big impact on your resulting estimate. And therefore, we've developed ways to decide what is the most appropriate heritability model to use for an analysis. But it's also important to realise that the heritability model is not just used in heritability analysis, but very widely in statistical genetics. In fact, most tools start by assuming all SNPs are equal, i.e. assuming the GCTA model. And so all of these tools can be improved simply by replacing this with a more realistic heritability model, as we've demonstrated for the case of prediction. So with that, I'd just like to thank the people who have helped me with this work, David Baldwin, John Holmes and Chen Cheng Zhang. I'd like to thank the people who have provided me with funding. And I finish just by pointing out that tools to perform all of these analyses are available within the LDAC software package, which you can obtain from www.ldac.org.